We're now going to look at the data and the stories of food, and I'd like to invite Maya Raja Sheikh Karan, the Managing Director for Africa at the Alliance of Biodiversity in SEAT, and Gina Kennedy, the Director of Research for the Periodic Table of Food Initiative with the Alliance of Biodiversity in SEAT, to join me on stage. And before they get a chance to speak, I'd like to welcome Jessica Prenny, an associate professor in the Department of Horticulture and Landscape Architecture in the College of Agricultural Sciences at Colorado State University, to give a short presentation and sort of set the stage for this next panel. Welcome her, please.
Thank you so much. So I'm going to actually talk a little bit more about data. So we heard a little bit about the tools, the standardized tools, and we saw that great demonstration of the interface that we can use to visualize that data. So I'm going to start to, I want to talk to you a little bit more detail about the foods that we analyzed and also some of the stories that we can start to tell with this data. Okay, so let's start with the foods. So we've heard this already. We've analyzed 500 foods, but what does that mean? Let's break it down a little bit. This included 357 plant foods, 59 animal products, and that included both animal protein and also dairy products, 32 aquatic foods, including fish, bivalves, and seaweed, 7 fungi and 1 bacterial-derived food, and 44 complex foods. And this is comprised of complete meals made for us by Chef Alejandra, as well as some commonly consumed processed foods and snack foods. Okay, and there we go. So importantly, this collection of food includes individual foods, right? So this is what allows us to start to investigate the differences between foods of the same type. So for example, the red apple versus the green apple. We've also separated out edible components of food. So in this example that I'm showing you here, we have a beetroot, and we actually separated out the beet greens and the beet roots and analyzed them separately. And then for foods that were not commonly consumed raw, we also cooked them. So this is showing an example of rice, which we analyzed both raw and cooked. Okay, so my number is a little bit bigger, but using the standardized tools, if we think about all of them, we actually have detected 21,110 unique biomolecules across all 500 foods. And this number includes all the molecules, so it includes the lipids, the minerals, the carbohydrates, and also those small organic compounds that Stephen Shiming just told us about. So with this data in hand, now we can start to zoom in and ask really specific questions about the biomolecular composition of a given food. So what is in this apple? In this apple, we detected 3,513 unique biomolecules. And what I'm showing you here is just a subset of these molecules, and this is a different way of looking at the data. And we're really focusing here on polyphenols. And so polyphenols belong to a class of molecules called secondary metabolites, and we've heard about these already. These are the compounds that plants make in response to stress. But there's also a lot of evidence that those compounds are good for us when we eat those foods, right? But these are not essential nutrients, so they wouldn't be captured in our traditional food composition analysis. But the goal, one of our goals of the PTFI, right, is to extend our measurement of these non-essential nutrients that could have impact on human or planetary health. And so I've highlighted a few of these compounds in this example, rutin, epicatechin, and chlorogenic acid. So these are specific polyphenols that we detected in apples that have been shown to be powerful antioxidants and have anti-inflammatory properties. And so with this zoom lens, we can also look at individual biomolecules. So now if we focus on this one polyphenol epicatechin, which we did find in the highest levels in apples in our 500 food set, we can see that we also detected this compound in other fruits like blackberries, cherries, plums, raspberries, grapes, kiwis, and cranberries, but also in some legumes like brown lentils and black and pinto beans. Right? So now with this type of information, we could start to think about how we might be able to tailor diets for specific bioactive compounds while also incorporating choice based on what fruits are available to you or which do you prefer to eat or are culturally relevant. Okay, so Now we can also zoom out, right? So we can zoom out our lens and we can look at this data across foods. And I know as a mom, like all of us as parents have probably told our kids, you got to eat your green vegetables. And we kind of tend to think of these green vegetables as all very similar. But in fact, what we're seeing here in this data, that they are not very similar. And so what we're seeing here, each of these bars represents a different green vegetable in our data set. And the height of the bar represents the number of unique biomolecules detected in that particular food. And in fact, of the 5,000 unique compounds that we detected across these green vegetables, only 202 were in common among them. So this really shows us that, in fact, these green veggies are more unique than they are the same. So in addition just to thinking about the overall number of biomolecules, we can think about the diversity of these biomolecules, which is often related to the overall quality of a food. And so here I'm showing you some data for some of our complex foods, right? So we have our fast food burger, and then we have a couple of alternative options that were prepared for us by Chef Alejandra. And so in this plot, I don't think I have a pointer. Do I have a pointer? I don't have a pointer. But the little bars in the plots represent the number of biomolecules in each of those foods, right? So we can see right off the bat that the lentil burger and falafel wrap are more complex in terms of the number of biomolecules we detected than the fast food burger. But those center sort of flower-looking plots can be interpreted. So in those plots, we have grouped the biomolecules by type, right? So that gives us a measure of diversity. And those can be interpreted as the further a point is from the center of the plot, the more of that, in terms of abundance, of that biomolecule group we detected in that sample. And so what these plots tell us is that not only is the falafel wrap more complex than the fast food burger in terms of the number of molecules, it is also more diverse in terms of the types and abundance of biomolecules. Okay. So finally, I also want to touch on this concept of dark matter that's been mentioned already, right? We don't know what we don't know. And this group of compounds, this small organic compounds, which Steve talked about and Qiming talked about, provides us with the biggest challenges in doing this type of analysis, but also perhaps the biggest opportunities for discovery, right? So of the close to 19,000, I can't remember Steve's number, 18,000 something, compounds that we detected in this class, only about 2% of them can be named as specific compounds, right? So there's a whole array of these compounds that we know are there, but we don't know what they are. And I can tell, so I've been working in this field almost my whole career, and this was not at all really an unexpected result. There are lots of challenges that make this process of identifying these types of molecules really, really challenging, and that is compounded exponentially in food because of the vast diversity across food types of these molecules. But I can say also that the PTFI approach is going to dramatically accelerate our ability to characterize these molecules. And so I'll just show you what I mean by showing some of these plots that Steve already showed. But just to walk you through it a little bit more. So this is an example of a plot where, again, all of the 500 foods are along the bottom, and they're color-coded by food type. And then the height of the peak represents the abundance of this particular biomolecule in each food. Now, in this case, this was a compound, one of our internal standard compounds. We added it to every food before we analyzed it. So as expected, we see it and we detect it in every single food. But in contrast, if we see a plot like this, right? So now we're seeing, this is one of our unknown molecules. It was detected reproducibly across 3 different laboratories, and it was detected only in soy-based foods. So this included edamame, tofu, soy milk, and tempeh. Right? So this consensus of detection across laboratories and across foods of the same type significantly increases our confidence that this is a real thing and that it is originating from soy-based foods. Right? So to put this into context, and this has already been, there we go. Okay? So to put this into context of how we're doing research now in this area versus the PTFI approach, right? So right now, researchers all around the world are doing research. They typically analyze one type of food. They generate their data, they write a great scientific publication, and that's kind of where the process stops, right? So even if that data goes into a database, a public database, the data was not collected in a standardized way, so it really impacts the value of that data for future discovery. Now, with the PTFI approach, we are enabling researchers across the world to analyze samples in a standardized way. They're going to analyze lots and lots of different foods, and they're going to generate data that's going to be standardized and comparable and be present in a common platform, right? So the PTFI database. And so hopefully these researchers will also still present their results in a scientific paper, but now the impact of that data is going to be dramatically amplified, and will fuel our ability to characterize these novel biomolecules in a way that we really just couldn't do before. And so these are just a few examples, because I only had 10 minutes, of how we can start to think about this data and use this data to tell stories, but hopefully I have highlighted how these tools and data can, or inspired you to think about how they can be used to answer or address these pressing challenges in human planetary health.
Thank you, Jessica. Round of applause. It's like you're doing deep space exploration. And I love it. Like when you talk about the impact of data, we just don't want it to sit in journals or in papers. We want it to be used in broader ways. So thank you for all of that. So Maya and Gina, I want to turn to you both now. And we've heard a little bit about the why of the PTFI. I didn't know I was going to rhyme there, but I just did. And I'm wondering if you can both speak about the why of this a little bit more, and why it needs to exist, and why all of these people are coming together to make sure that it happens. Do you want to go first?
Sure. First, absolutely delighted to be here. And I want to talk a little bit about the part of the story of the food as well. And I think, Jessica, you mentioned about 500 food. Most of these foods come from United States. And part of that story is also like where these foods come from. Mm-hmm. So from the very beginning, when PTFI started, we spent quite a lot of time thinking about, do we have the permission to access these resources? Do we have the permission to actually put some of this data out there? Who can actually use it? Who has a capacity to use it? So the benefit sharing was very much in our thinking from the very beginning. And so Food EDU and Good Food Fellow, some of that really comes from the thinking about like, how do we make sure that this phenomenal resources that PTFI is putting is actually having a benefit in equitable benefit sharing across the different parts of the world. So food is a biological resource, and they are regulated. Its access and the information derived from this food is regulated by international treaties, and some of them are called in Convention for Biological Diversity, Nagoya Protocol, and few others. So we actually spend a significant amount of time to really making sure that we have actually the right and the permission, and we really work with the national agencies or other, really taking help from people who really understand how do you navigate this international protocols and treaties, and really making sure that before we put out the data out there, we really have the permission to do so. Because these are highly regulated, and it's really important for the PTFI to make sure that we do the right thing. So PTFI, from the very beginning, took a very proactive approach to access and benefit sharing, and it's not looking only at the monetary benefit, it's really looking at, okay, we can provide benefit sharing in the form of capacity building, and that's Food EDU and the Good Food Fellow. We can do the benefit sharing in the form of partnership. So you see the broad spectrum of partnerships that are coming from all parts of the world, and we can do benefit sharing in terms of really putting resources to address some of the key questions and having a deeper exploration of the data and asking some of the research questions. So PTFI took a very broad spectrum approach to access and benefit sharing, at the same time really making sure that we can actually put some of this data out. Sure. Chiming really showed that the food data, we have 4 different levels of data sharing permission. That level 1 is like, it can be publicly shared, it has no restrictions. The level 2 is basically that we're still checking, we're not sure, we are still working with the national agencies to see whether we have really made sure that, we access a particular, let's take an example of a teff from Ethiopia. Mm-hmm. We really want to make sure that we have the right permissions to use that, or from Mexico, a particular crop. So some countries does not regulate the access and use of these resources or the biological resources, edible biodiversity, but many countries actually do. And it is a very, very complex issue and topic. And PTFI is actually, with the help of Rockefeller Foundation, is also proactively engaging in conversations like DSI, digital sequence information. So Rockefeller has convened certain community and conversations to really say that how can we shape these global conversations, and we need to do the science, and we see that benefit of these amazing innovations, but how do we actually shape some of these global conversations in a way that science can go forward, but at the same time really work with the countries and the regulations. So just to stop on that, we have 4 levels of different data permission. Level 1, publicly available, anyone can access it. Level 2, we are still checking. Level 3, level 3, we make sure that, okay, after checking in level 2, we decided that, okay, it's going to level 3, we cannot use it. Level 4 is also very much like we talk about indigenous communities. Many of the resources, traditional knowledge associated with communities, indigenous food are highly regulated. And so we really want to make sure that this is done extremely properly. We go through all the right steps. Even if it's a long steps required, we really want to make sure that we do the right thing. So that principle of equitable benefit sharing and really making sure checks and balances, informed consent is part of the story of the food is a major part of that database and some of the thinking that PTF has gone through in the last couple of years.
So incredibly important to have those rights and permissions in place so people are not taken advantage of, and that farmers and indigenous folks are not taken advantage of. Thank you for going through all that. So important. Gina, can you talk a little bit about that why of the PTFI and why we're all here?
Yeah, absolutely. Thank you, Danny. I'm so thrilled to talk about the research that the Periodic Table of Food Initiative is embarking on now, that we have these amazing standardized tools and this ability now to put the data into the database. So we have 3 really overarching questions in the research program for the Periodic Table of Food Initiative. And one of those is what's in our food? And so the foundation of that, of course, is the world's food biodiversity that we've been talking about all day yesterday and also today. And it's just a thrill for me to be part of a project that actually wants to explore foundational research about what is in our food biodiversity. And with the ecosystem that's already set up, with our 9 centers of excellence, we can add to what we already have about these 250 species that mostly were procured from the United States. We're going to add to that thousands of fascinating other foods. We have this list of 1,600 foods that we're so excited to explore. So for example, what is in nopales? We're going to get a really deep exploration of nopales and what role does that have for human health and nutrition by looking also at human serum and biomarkers. We're going to look at tropical fruits in Colombia, chontaduro, and I'm mispronouncing it, lula. We're looking at fungi, mushrooms in Thailand. We're looking at nama, or sea grapes, in Fiji, and ota, river fern, in the Pacific Islands. All these things, and having so many great conversations with chefs, like Chef Alejandra saying, I need to go to the PTFI and find what is in nopales, what is in, Robert Oliver will say, I need to go and find what's in noma or sea grapes. So they'll be able to actually find their local foods because of how we've set up the global ecosystem to have our 9 centers of excellence. So what is in food is, of course, fundamental question. And then the next question is, how does that vary based on how our food is produced and how our food is processed? And that's when we also can get into some questions about what is possibly in our food that we might not want to be in our food. What are the concentrations of lead and of cadmium and of arsenic? What about antibiotic residues? What about pesticides residues? What about trans fat? We saw that fascinating graphic from Jessica about the fast food burger and then 2 different alternate types of burgers. That's something also that our colleagues at UC Davis are exploring. They're looking at the differences in the protein, the proteomics of different kinds of plant-based alternatives to animal source foods as well as animal source foods. So we'll have just so much rich questions that we're looking at with all of our partners. And of course, then the implications for this for human health and planetary health. So I guess just to say that the power of PTFI is really the power of our partnerships.
Yeah.
We're so thrilled to have with us our 9 centers of excellence. We also have national lab partners whose mandate is to look at food composition for their countries. So as Maya explained, it's a very country-driven, country-informed approach that we would like to take. So we are bringing on national labs whose mandate it is to develop food composition for their countries. And then we also have several other research partners at universities and academia, and also with the CGIAR centers. So we're just developing this great ecosystem, and we hope that also those in the audience and those listening online will be inspired to join us in our pursuits.
Absolutely. It is such a cool ecosystem, and I'm especially interested in what you'll find out about the different farming practices and processing techniques and how that affects what's actually in our food. It's really, really exciting. So thanks for sharing all that. Jessica, if I can come back to you. Sorry, I'm missing words today. Is there anything that surprised you about the research that you want to share?
I think the biggest thing that surprised us was, like Steve alluded to, is how many examples there were of molecules that were clearly coming from one type of food. And that's why we both spent hours and got lost in that data. But the power of that information to help us when we have data from so many different labs and so many different foods, to be able to help us characterize these unknown things. I think there's just so much there for us to discover.
Right. I keep using the word exciting. I've overused it the last 2 days, but it is so exciting.
Yeah.
It's like this whole new world that we didn't have this kind of access to before, right?
Absolutely. Absolutely.
These tools are amazing. So this is a question for all of you. There are so many benefits from the PTFI that cut across a bunch of different sectors and industries. How can it benefit, for example, we've heard a lot about food as medicine over the last 2 days, or regenerative agriculture efforts. Maya, do you want to take that on first in the last 3 minutes that we have?
Yeah, I think I would just take it from maybe from a CGI perspective. I mean, we have sitting in a huge, vast collection of gene bank resources, thousands and thousands of accessions. And the genomics have advanced so much, and phenomics have advanced so much, so it must be an incredible opportunity to bring that foodomics aspect, to really bring the data and look at the integration and see, the precision, in terms of breeding, in terms of what is possible, is amazing. And in the opening, we mentioned that we eat a very small number of crops and food from around the world. And there's a huge number of opportunity crops, a new initiative called VAX, Vision for Adaptive Crops and Soils. And PTFI provide a phenomenal opportunity to really look at these opportunity crops into much more details and look at what's there, how can we really, and there's a lot of data gaps there, so we have a phenomenal opportunity to really bring that forward. And also the link between agriculture and health. The CGIAR also advanced significantly in the last decades on really linking agriculture to nutrition, and this is a phenomenal opportunity actually to link that ag and the health into really one platform. So just from the CGIAR perspective, I see really amazing opportunities for us to build on the efforts of the PTFI and the tools and the data.
The opportunity is so big. Gina.
Yeah, thanks. I'm just going to build on what Maya was saying about the linkages and also Roy and the vision of those who started the Periodic Table of Food Initiative to combine health and agriculture sciences together. And so there's one project that I'll just mention. There's several projects, so I don't want to just mention one. This one is funded by the Rockefeller Foundation, and it's called Soil Health, Food Composition, and Human Health. And why it's an interesting example is because they're taking the Ionomics platform, which is a platform that's looking at 26 minerals and metals, some of which we need in human nutrition, say iron and zinc, and others you want to have it in lower levels, such as lead or arsenic or cadmium. And what this project is doing in Ethiopia and in Ghana is they'll take soil samples and they'll look at what are the minerals and metals in that soil. And then they're going to interview farmers, and they're going to say, what are the practices that you're using? What are you doing to grow this food? They're going to take the sample of the food grown in that soil. They're going to analyze it using the same Ionomics platform, so the same 26 minerals and metals. And then they're also going to, on the health side, interview people about their diets and take blood samples and take stool samples so that they'll know from the blood Do we see the same minerals and metals that from the soil into the food, into the blood, and also into the microbiome? So it's this real frontier science, and it's really, really bringing together agriculture and health, I think, in ways that haven't been done before. So it's super exciting.
That's amazing. Thank you so much for sharing that. We are sadly out of time. Let's give these panelists a round of applause, please. Thank you all so much. Bigger round of applause, everyone. Keep the energy up. These days are long.